Go.
The second option is cyclic electron flow. This is an alternative pathway that uses only PS1 and the first ETC to generate energy for ATP synthesis, but no NADPH or oxygen. This process starts when a photon of light strikes a pigment molecule in the light harvesting complex and boosts the electron to a higher energy level. This amount of potential energy makes the electron unstable, so it falls back to its ground state, exciting the next pigment molecule. Note that the electron doesn't actually move physically. This process continues until the P700 of the photosystem 1 is excited. P700 is then oxidized to P700+, losing its electron to the primary electron acceptor. The electrons are replenished when water is split, as explained before when talking about linear electron flow. The electron passes from the primary electron acceptor of photosystem 1 to the ETC, in linear electron flow would come after photosystem 2. The exergonic fall of electrons to a lower energy level throughout the ETC provides energy for ATP synthesis because, as electrons pass through, protons are pumped, building a proton gradient used later in chemiosmosis. These electrons are shuttled back to chlorophyll A, restarting the process. Next comes chemiosmosis. This couples redox reactions in the ETC to ATP synthesis. As the electrons are passed through a series of progressively more electronegative acceptors in the ETC, which comes after photosystem 2 in linear electron flow, or is the only ETC in cyclic electron flow, protons are pumped across the thylakoid membrane. Thus, redox energy is transformed to the proton motive force, which is energy stored in the formula of the hydrogen ion concentration gradient. ATP synthase couples the diffusion of hydrogen ions down their gradient to the phosphorylation of ADP to ATP. This is called photophosphorylation because electrons are extracted from water, not from organic molecules, as in oxidative phosphorylation. Overall, light energy is converted to the chemical energy of ATP. Basically, hydrogen ions diffuse through ATP synthase from the thylakoid space to the stroma down their concentration gradient to provide the energy to photophosphorylate ADP to ATP. 9 ATP are needed to fuel the Calvin cycle.